Hey Iqbal. Baik, selamat malam Bapak, Ibu, dan adik-adik semua. Suara saya terdengar ya. And good afternoon to Mark. It's, I think, 1400 in the Netherlands, right?
It is.
Um, Thank you very much, Mark, for agreeing to give the guest lecture today. And so the lecture is intended basically for the students of agricultural nematology course, but I've opened it for the public basically, and I see that some people already attend, which is not the students. I see some familiar names, which is a nematologist in Indonesia who are nematologists in Indonesia, so should be fun. Um, we are expecting at least 70 to 7 people as the number of my students, but I suppose some of them are joining screens, so like one screen are used by multiple people, one laptops or so. Um, so before we start, I don't know if Bu Siwi is here already. Hmm, maybe not yet. Okay. All right. So I think we can start, and then people will still come eventually, so no problem. So, first of all, I would like to thanks Mark for agreeing to give the lectures and also welcome to my class. Most of them are my students. And so today, Mark is going to talk about management of the potato cyst nematode in the Netherlands, a little bit about maintaining or managing population dynamics of Globodera in the field, and also what is the main or what is um the few most used methods to manage them, let's say. And before I give the floor to Mark, I would like to give a little bit information about him. And yeah, so I'll just show Mark's Google Scholar. As you can see that Mark is associate professor at Wageningen University, and his his field of expertise covers quantitative genetics, genomics, host parasite interaction, plant nematode interaction, virology, and Mark has published in multiple high-impact journals, very prestigious journal, including Trends in Genetics, Nature, Nucleic Acid Research, Nature Communication, BMC Biology, New Phytologist, BMC Genomics, and so on. There are so much. I'm not gonna say it one by one. And also, Mark is currently supervising and already graduated no less than 10 PhDs, I believe, and I'm honored to
Correct.
be one of the PhDs that he supervised and um, yeah, mentored under him. Um, in fact, I was one of his first PhD students, which I'm very proud of. And yeah, so um, Mark is now Mark also received multiple very prestigious grants, including FENI and FIDI, and also multiple European Union grants, in which then he starts his research groups and in the genomics of plant parasite and plant nematode interaction. So that's a little bit introduction of Mark from me. Without further ado, Mark, I will give the screen to you and you can start sharing your screen and presentation. So the idea is that we're going to have like maybe 40 minutes, 45
Yeah.
minutes max of the presentation. And then after that, we're going to have a Q&A. And I would
Yeah.
really encourage my students to ask questions to Mark and also for other people as well, other nematologists that are not students anymore. You can also ask questions to Mark later on, so feel free. All right,
Yeah.
Mark, screen is yours.
Thanks for the nice introduction, Iqbal. Indeed, Iqbal was one of my PhD students, and he graduated a little while ago. He wrote this very nice book, as all Dutch PhD students do. So, and we're working on publishing that. I think we will do soon.
Yeah.
But yeah, Iqbal, thanks a lot for inviting me. It's a pleasure to talk to you and your students. I think you can now see my screen
Yes.
with the presentation. Great.
Yes.
I have
Yes.
a
You're
pointer
saying.
ready, so this should all work. Yeah. So my name is Mark. Please just feel free to call me Mark. Everybody does. And I want to talk to you about the management of Globodera or potato cyst nematodes in the Netherlands. And I will talk in particular about Globodera pallida. I will touch a little bit upon Globodera rostogiensis. So maybe you know, maybe you don't. The Netherlands is a bit colder than Indonesia, but I think just as wet. So this is the state of a potato field in the Netherlands near winter. As you can see, it's quite muddy. The potatoes have been partially harvested. So on the left side of this picture, the potatoes are still in the field, and on the right side, they have been removed. And you see maybe some people walking in the background. Those are three colleagues of mine with whom I went to this particular field to sample Globodera pallida. So nematodes, of course, cause a lot of damage. This is beet, but the type of damage is quite similar. What you see is that plants grow a lot slower because they cannot take up so many nutrients. And for the rest, there is no very clear visible sign of damage. Maybe you see some leaf yellowing. However, the point I want to make is here is that for farmers, it can often be very difficult to recognize the symptoms of nematode infestations. And the reason is that these types of infestations also look like nutrient deficiencies, especially earlier on. So if you don't have high density of nematodes, then this can really simply look like a lack of, for instance, nitrogen. Of course, we as nematologists, we know what is underlying these kind of symptoms. So we have nematodes that actually really like plants. They like plants in a similar way as us. They like to eat them. So what these nematodes do, they go towards the plant and they infect them. And the species I will talk about, Globodera, it's a specialist. So it specializes on solanaceous plants, particularly potato, though they can infest tomato plants as well, but not as good as potato. So the goal of what I'm going to tell you today, as by request of Iqbal, of course, is about how we actually manage these species in the Netherlands. And I want to discuss three things. I want to discuss plant parasitic nematode management strategies in general. Then I want to discuss resistance breeding in particular. And then I also want to look at how policy and mitigation strategies are shaped in this species, which is mainly focused on pattern types. So let's dive in. So first, I want to discuss nematode strategies in general. And for that, I want to start with the life cycle of plant parasitic nematodes. Maybe you know, I guess you do because Iqbal taught you, but a bit of repetition cannot hurt. Plus, I will talk about the particularities of potato cyst nematodes. So if we start at number one, hatching, potato cyst nematodes hatch mainly because they sense that there is a potato plant nearby. They sensed it because plants are leaky, their roots leak, they excrete what we call root exudates, and nematodes pick these up, and potato cyst nematodes pick up exudates of solanaceous plants, and that induces hatching. Once the nematodes hatch, they start migrating towards the plant, and then they have this little needle-like structure on the top of their head that they use to destroy the plant root and to enter the root system. This is a bit different from Melidogyne. So Melidogyne will seek the root tip and enter through there, whereas cyst nematodes like Globodera, they will actually destroy the root and use damage to enter the root system. Once these nematodes have entered the root system, they will start developing a feeding site. This is actually quite a complex process, which involves the nematode producing all kinds of proteins and other things to manipulate the plant cell to form a feeding site from which they can feed. So this is really a complex interaction between the plant and the nematode. Then at some point, these nematodes start to reproduce. So you have males and females. And ultimately, the females will form the cyst. Cysts are visible by the naked eye, and these cysts can contain approximately 300 eggs, and they can survive for a really long time, well over a decade, and it has been reported for up to 40 years of survival in the soil. So this makes it really difficult to eradicate this type of pest. So these Globodera species, they do not come from Europe. They were actually introduced into Europe, and we think around the 1850s. The exact date is not known. It's estimated to be somewhere between 1800 and 1900. We do think they were introduced when, ironically, when people were breeding against another potato pest, namely Phytophthora infestans. So this famously caused the Irish potato famine, and around that time, expeditions were organized towards South America, and we think that these expeditions brought along Globodera pallida and Globodera rostogiensis into Europe and into our potato cultivation industry. This was not noticed at first. The first reliable reports about Globodera in Europe were past the 1900s. So the first one was in 1913 in Germany, Rostock, from which also the name rostogiensis derives. And in the Netherlands, the first reliable report was in 1938. And we think that these nematodes were present earlier on. But yeah, there are simply no reliable reports. So we actually know quite well where these nematodes come from. Genetic research has pinpointed their location of origin near Lake Titicaca in the south of Peru. And it is pretty clear from genetic analyses that European populations and probably also global populations, because we think all the secondary spread around the globe came from Europe. We're sorry for that. If you have pallida in Indonesia, it probably came from Europe. But yeah, it is genetically very clear that this species comes from there. And the reason is also that potato has its center of diversity also around that area. So it's also a very logical place to start looking for potatoes to breed with. Recently, it was published actually for Globodera rostogiensis that indeed this secondary spread was facilitated by Europe. So also here, the populations originate from the south of Peru, then were introduced into Europe and spread along. So what you actually can see is an analysis of microsatellites. And you see that in the south of Peru and the north of Bolivia, you have this red genotype. But in the south of Peru, you have this blue and yellow colored genotype. And then what we think happened is that this blue and yellow colored genotype went to Europe. And secondly, secondary spread from there, let it go to Africa, Indonesia, and America. Because that's the most parsimonious explanation for what happened. Of course, we recognize these problems in our agricultural fields, and then people, of course, start to work to actually deal with these problems. So people started breeding for resistance against these nematode species, and that led to the discovery of, I think, the most famous resistance gene in
Iya,
all of nematology, namely the H1 gene. This was discovered in the 1950s, was published in 1952 by an English and a Dutch group. And it was also soon brought into commercial use. The first resistance races were introduced in the early 1960s, and in the Netherlands, these were Amil and Amilio. And from that, actually, this gene has been incorporated in basically every Globodera rostokiensis resistant cultivar that I know of. So it's very ubiquitously present. You find it all across the globe. People are using this gene to control Globodera rostokiensis specifically. This discovery had a side effect, which I think I will touch upon later, is that actually it led to the discovery of Globodera pallida, so the second species. And the reason was that this species looks morphologically very similar. If I look at them under a microscope, for the life of me, I cannot distinguish these two species. But actually, they behave differently on this H1 resistance. It controls Globodera rostokiensis very well. It does not control Globodera pallida. And that then brings me to management. So the management of these species is governed by a legal framework here in the European Union. Before we had the European legal rules, we actually had a law in the Netherlands for the control of potato cyst nematodes. I think it is quite similar in Indonesia. This is a quarantine organism. I think it is globally considered a quarantine organism. And that means that the goal for the legal goal for this organism when you detect it is eradication. Well, if we translate it then to practice, we scientists, we do not believe we can eradicate Globodera. This is not possible, simply biologically speaking, economically speaking, it's just unfeasible that we can do it. Therefore, we advocate and we think that management is key. In other words, we have to be very aware where we have infestations, so which fields are infested, and we have to locally manage the populations. So we have to suppress them, make sure that they don't spread, and in that way control this pest. The key here is that the places in the Netherlands where seeding potatoes are grown, so the potatoes that are then sent to farmers to actually grow the commercial crops, these have to be really, really clean. Like there you should have no infestations at all. Because if you start spreading around this pest with seed potatoes, you're done. You get it everywhere. But there are particular commercial areas in the Netherlands where we have these pests, and we simply cannot remove them from there. This is economically impossible. So we have certain tools to actually establish management. There is monitoring. There is both monitoring by the government, which is legally required, but also there is voluntary monitoring. So farmers voluntarily let their fields be sampled so they know what is going on. Then the most important control measure next to resistance genes is crop rotation. So you don't grow a single crop all the time. You rotate different crops as far as you can get your building plan as a farmer round. So that means, can you make enough money by rotating through a couple of crops? Then there used to be a lot of nematicides. We had chemical soil fumigation, which was really environmentally hazardous and also very toxic for humans. That is completely banned now. So that was methyl bromide. I think it's banned in most countries nowadays. There were some nematicides that were still allowed, but actually the most potent ones have been banned 2 years ago as well because of environmental concerns, meaning that we have very little chemical control currently. The most important management tool, therefore, is resistance genes. So let me first go into crop rotation to show you the impact on nematode numbers in the field. So this is from simulations, but it's quite well known what the effects are, because it's been extensively studied what happens with potato cyst nematode numbers in certain rotations, how much biological decline there is over generations, etc. So let me start with a 1 in 3 rotation. So we start here with a certain number. The starting year is 1996. And then we grow a susceptible crop. So this has no resistance genes at all. It's a very simple scenario. We have a potato that we grow, no resistance, simply a potato. You see that the numbers increase. And then if we don't grow a potato the year after and the year thereafter, we see the numbers decrease again. We know that in the first year after growing a potato, the decrease is 50%. Every year after that, the decrease is 33%. We know this very well because people have painstakingly measured this over decades, actually in the field. So, I mean, there is some variation around this number, but we're pretty sure that this is the decline. If we then grow a potato again, the numbers shoot up, because there are still all these cysts in the soil. They will go into the root, they will multiply, and we get more and more cysts. Then it will decrease again, but with a 1 in 3 rotation, you see that you go to this maximum, because there at some point there can simply not be more nematodes in the soil. And for the Netherlands, in sandy soils, we know that this number is approximately 150,000 juveniles per 200 grams of soil. Then after that, no more nematodes fit. That is simply it. So if we go to a 1 in 4 rotation, you see that you get a bit more decrease, but still you go to this maximum number over time. So the x's are all the same, and you see you end up somewhere here in this simulation with 80,000 eggs per gram of soil. However, if you were to 1 in 7 rotation, you actually see that over time the populations decrease. So in other words, if you grow a 1 in 7 rotation potato, you will not, over time, you will actually drive the population to extinction. Because, and this will take a long time, but ultimately it will happen. So we nematologists, in the sense of potato problems, we would not have work if people were growing potatoes 1 in 7. So this, however, is economically unfeasible. We grow in the Netherlands, we grow potatoes at industrial scale. There are large factories processing these potatoes. And in some areas, 1 in 3 rotation, 1 in 4 rotation works. And in some areas, even people grow it in a 1 in 2 rotation, which is really, really a lot. But there is no other feasible economic model possible. So only on biological farms, farmers will not have serious potato cyst nematode problems, because they can actually grow 1 in 7, 1 in 8. Then you're completely fine. You don't need resistance to control this particular nematode species. Of course, it's a very different story if you have Melanogyne incognita or something, because it just, affects many, many different types of plants. So this is how we think about management in general. And then I want to go to resistance in particular, because that is one of our most important management strategies that we have. So I want to address 2 key questions. One is, what is a resistance gene? And second, what is virulence? Because these 2 things are important terms to know in terms of management of nematodes. So resistance genes have been cataloged for potato cyst nematode for a long time. I think the first resistance gene published was the H1 gene, as I showed at the beginning. And ultimately, also this gene was it was pinpointed on the potato genome where this gene resides. People made molecular tests, so PCR tests, to track the gene in breeding material. And this was not only done for the H1 gene, this was done for many different resistance genes. So this is a nice review from Ulrike Gardner, listing all the potato resistance genes known to date. I say genes, but you actually see that they make a distinction here between genes and QTLs. That means that for these three, we actually really know the gene. We know the actual sequence in nucleotides on the genome. So we know that it starts with the ATG and then all these other nucleotides after it. However, for most of them, we only know where they roughly are located in the genome. So we don't know actually which gene is causing the resistance. We just know, well, if you roughly have this piece of potato genome incorporated, you test this by PCR, then the plant will be resistant, most likely. We do, however, know quite a lot of how these resistance genes work. So they are monogenic, typically, so single gene, and it's a very black and white phenotype. In other words, your plant is either resistant or it is not resistant. So this resistant controls a particular nematode, typically a particular nematode species, and sometimes even a particular nematode petotype. Especially from fungi and oomycetes, we know a lot about how these genes work. So they are what we call ND-LRR proteins, so nucleotide binding loose enriched repeat proteins. I don't know if you have to know this for your course. I guess not, but in essence, what you can view them as is this loose enriched repeat as sort of a sensor. So this is typically varying between genes. So there's a particular sequence, and this particular sequence can recognize something that has to do with the pathogen. And then you have this nucleotide binding, and that is typically one of the parts that ultimately leads to signaling. So it will then do something in the plant cell, activate programmed cell death to get rid of the pathogen. If you look at these in the context of the potato genome, we see that many of these NB-LRR genes, these potential resistance genes, cluster together. So you have hotspots on the potato genome that contain many different resistance genes. So that is not only against potato cyst nematode, but also against Phytophthora infestans, warts disease, and all kinds of other potato diseases. And again, this is a general pattern. So you also would find this in tomato, for instance. Also here, resistance genes tend to cluster together on the genome. On the one hand, that is sort of nice, as if you will become a plant breeder, for instance, and you know that, well, often these genes are sort of residing together. But this can also be a little bit tricky, because it means that if you want to combine resistance properties, it can be difficult from a breeding perspective, because they might be on different pieces of DNA. So they might be close physically speaking, but genetically speaking, they might be on different pieces of DNA. And in potato in particular, you have to think of that it is a tetraploid. So it has four DNA strands per location. And so tracking all of that, combining all of that, it is quite some work. So I mentioned that typically we do not know the gene. We know a quantitative trait locus, a QTL. So this is what that means. It means that we know by PCR that there is a particular place on the genome associated with the trait, so being resistant. For instance, here is GPA5. And I highlight GPA5 because it is a very important resistance in controlling Globodera pallida. However, we do not know the gene. We just know that there is a piece on the genome that correlates with being resistant. And this gene in particular has been used a lot. So this is a supplemental from a recent publication of ours where we show that actually this GPA5 gene is used in most resistant cultivars grown in the Netherlands. And actually, we think it is used in most resistant Globodera pallida resistant cultivars in Europe. To be very precise, we think it is the cornerstone for Globodera pallida resistance. So this is the most important gene currently in use. And as you can see, we between the horizontal lines here, this is the area where we think this gene resides. Because again, we don't know the sequence. But we see that all the resistant cultivars, and that is everything you see in this figure except this red one called Desiree, they all have some variation, genetic variation in this area, that's highlighted in green. So they all have the potential to have this GPA5 gene. And this is also the best known location there is. We simply do not know it more exactly than this. But it is used a lot. And so what it does, it is controlling a specific petotype. So a petotype is simply a definition. So we say there is a PA3 petotype, and that actually means this is controlled by this GPA5 gene. So the definition of PA3 is it cannot replicate well with this resistance. And there are two PA3 lines used in the Netherlands. There is the E400, which is a Dutch population, and Chavornay. Chavornay, this population comes from Switzerland, and it is the European legal definition of PA3. So it is anchored in law that this specific population is PA3. And what you can see is that it cannot replicate very well. So this is a relative replication, and you see that it is below 1%. And that is, again, is simply a definition. There was a definition made in literature in 1977 that replication under 1% constitutes resistance. Here I have two recent field populations recently isolated from the Netherlands, and you see that their replication percentage is a lot higher than this 1%. And that is what we call virulence. So virulence is the ability of a population to break the resistance. And in Globodera pallida, that happened because there was a copy of a virulence gene in the genome. It was not present a lot, but it was present in a few percent of the population that allows this nematode to grow on this resistance gene. So let me show you, let's translate this a bit more to practice. And also want to prepare you a little bit, because in a little while I'm gonna ask you to do a little exercise, so you also get a bit familiar with how we interpret these petotypes. So you will need maybe a little like a pen and a paper at some point. So the effect of resistance is that you can actually grow potato in a higher frequency. So if you really look at it agronomically, so maybe you remember I showed you what happens if we grow potato 1 in 4 generations, 1 in 4 years. So if we don't use a resistant cultivar, the population increases when we grow potato, then declines a bit, then increases again, till we get actually a lot of potato cyst nematodes in the field. However, if we grow a resistant cultivar, this is what happens with the population. So it stays really controlled at low numbers, and there is no, initially at least, there is no agronomic damage. If you pay good attention, you see that there is a little bit of an uptick here. And that is because this model I run here takes into account that there is selection of virulence. So at some point, the resistance stops working and the population can grow again. So I hope what you picked up from this to get you on the same page again is we have nematodes in the field. We cannot grow our targeted crop in such a low frequency that we can rely only on crop rotation to control them. Therefore, we use breeding for resistance to control nematode populations. However, these resistances can be broken, and we call that virulence. And also, these resistances, they are defined as, they also drive a definition of a pathogen called a pathotype. And these pathotypes are really key in shaping policy and mitigation strategies. And that's also why I want to highlight these a bit. So we're going to do a little exercise. I'm going to explain it to you, and then I will give you a bit of time to do it. Okay? So the exercise is to build your own pathosystem. So here I have a couple of fields. So give them a little bit of different green colors. With every box you see represents a field where we will grow potatoes. And in these fields, we have 3 different populations of our nematode. So we gather these populations, and we, of course, also have 3 types of resistant plants. So we call them resistance 1, resistance 2, and resistance 3. And so we have population A, B, and C. Then I will give you a couple of facts. So you did some experiments, you added the nematodes to your resistant plants, and you started looking whether or not the nematode can actually grow on this particular resistance. So this is the information you get. I will do the first example with you, and then you have to figure it out on your own. So the facts are: population A does not grow on plant 3. Population B grows on plant 3. And then information like resistance 1 propagates A and C, but not B, etc. I think you can read, so that should be fine. What you should construct is a little matrix like this. So we have A, B, and C in the rows, 1, 2, and 3 in the columns. And then, for instance, you can derive that of plant 1 propagates population A. So you fill in a yes. Then also we want to fill it in for population 3. For resistance 3. And population A has no growth on plant 3. So here you would fill in a no. So with that information, try to finish this matrix. And then the question is, how many pathotypes are there? And please write your answer in the chat. I think I give you 5 minutes max. We see when the answers dry up.
Oke, adik-adik silakan dikerjakan ya exercise-nya. I'm just telling them to do the exercise more.
Yeah. It's good exercise for them, right?
Yeah. Nice exercise.
Will you also make it, Iqbal?
Yes, I'm also working on it.
Good. Don't be shy about adding an answer. It's completely fine.
Yang sudah mengerjakan bisa ditulis di chat jawabannya berapa gitu. Satu, dua, atau tiga, atau type. Someone says two with a
Yes,
question mark. She's not sure.
that's completely fine, huh?
Yeah,
Someone agrees.
someone agrees.
Many agree.
Many agree. Hmm.
Yeah. Thank you for answering, huh? And putting your thoughts in. Ooh, someone first supporting, then withdrawing, then supporting.
Okay.
Yeah. So thank you for that. I think it is. I think this is a useful exercise to do, and I will say to you that there is not really a completely perfect answer, though. I think you can argue for two, but yeah, it has some drawbacks because you don't have actually complete information. I think that is also Gali refers to, because yeah, we lack information for resistance to and pattern in population B. But I think this is actually also the point. So we can still sort of say well, we can sort of still argue that there are then two pethotypes. And in this case, 1 and 3 we would call differentials. So those two populations at least give us a yes and no answer that divides the populations up. But actually, resistance 2 can have additional information. And maybe if you have more populations, you would arrive at different answers. So that is my whole point with this exercise. Because you say 2 question mark, and I think that question mark is actually really important, because in reality, we will not have full information. You cannot measure every population, every field. The thing you can do, you can measure one population, find a particular answer, 2, 3, 4, 5. You can do quite a bit, and you can become more confident about your answer over time, but you will never fully know. And for potato, it was, so you now did it with 3 by 3. This is the answer for potato. At least the answer from 1977. So this is what people tested. So here, the nematode populations, they are here now on the columns, and the potato populations, they are here on the rows. So it is flipped. But yeah, these hybrids of Solanum tuberosum, these hybrids of potato, they are what we call the differentials. So they are plant 1 and 3. And these are all different populations. And you see that some were from the Netherlands, Dutch, some were British, some were German, like Harmerts. And I think by just solving this 3 by 3, you also appreciate what kind of a puzzle it was to get to this. I don't even know, 7, 8 by I don't know how many. I never really actually count them, because I don't really count them, because this is, how do I put it? It's a legal reality. So it's true in the sense of the law, but scientifically, it is not the truth. Let me explain. This plus, it means that this particular population, row 1, it can grow on potato. Fine. It can also grow on this particular hybrid. This minus, it doesn't mean 0. This minus, it means that the growth is less than 1% compared to a fully susceptible potato. So there can still be population growth, just not a lot. So when this was published in 1977, it was immediately criticized. Yet, it became anchored in law. So if we look at the Dutch Plant Protection Service, and I'm sorry this website is in Dutch because it's for Dutch farmers. But if you were to look at this website, you will find that they are testing for these standard populations: PA23, row 1, row 2, 3, and row 5. So actually, every potato cultivar in the Netherlands that can be grown to combat potato cyst nematodes, they are in this list. So this list contains, I think, almost 700 potato cultivars. And all of these potato cultivars, they get a score. So they get a score depending on how much relative propagation they have. So for instance, you see here that most for this row 1 population, most score a 9. There are a couple of lower grades. Then for row 2, 3, they have a certain score, etc., etc. And this is actually what is then used in practice. So a farmer can have a nematode problem. Then legally, this farmer is required to take a countermeasure. And one of the legally allowed countermeasures is growing a resistant potato cultivar. So this farmer can say, oh, I have Globodera pallida in my field, and I actually want to reduce it because I have to control it. So the farmer will go to the website of the Dutch Plant Protection Service and will look at a potato cultivar that can do this controlling. So, for instance, you can take BMC or Vestine. They score a 9 for pallida, meaning the propagation is really low, not zero, low, and they can grow this this potato cultivar. The way that these numbers are derived, there's an evidence-based framework for this. So it is governed by the European body for plant protection, the EPO. And to get a grade, a number in this system, you have to do 2 consecutive years of 5 replicates of 2-liter pot tests, at least in the Netherlands. So meaning 50 for your potato cultivar, you have to have 5 pots in a certain year, to which you add 10,000 larvae, and you count the number of eggs and larvae produced after 14 weeks. And then you have to do it another year. And then the average of the propagation after these 2 consecutive years is the number that ends up here in this list. This is quite expensive, but because it is a legal requirement, companies can, of course, make a profit with this. And this pathotype scheme, it affects the breeding, because so you see here all these nice gene and QTL names, but you also see for which pathotype they control. So every resistance is coupled to a pathotype. So what I call this flawed method being translated into law also makes that it is being used still as a breeding target. So I mentioned this timeline. So after these discoveries and control, because there were many problems. Actually, the department I work at and where Iqbal got his doctorate, we were founded to control this particular species. So that is the Dutch government gave money to Wageningen University to start the Department of Nematology, specifically to combat potato cyst nematode. So we have many populations. We gather these, and we saw that initially this H1 gene, it worked really well, but then we got a pallida problem. And later on, also in the 1980s, we got even more problems. Also, we found that there are other arguments why this pathotyping scheme is not the way to look at these species. I think that goes a bit beyond what I'm telling you today. But if you are curious, I encourage you to think about it a bit, and maybe if you can explore some of these links. In essence, we had a good control with this GPA5 from Vernin, so the gene I've been talking about a lot. But actually, since the 2010s, we have new problems with virulence. And that the basic reason I talked a lot about is that the propagation is not zero. It is like maybe 1%, but this 1% has a genetic basis, and it's selected over time, and you get a population that can grow very well on your resistant cultivar. So, in other words, this virulence, it is relative. So we try to talk about it in black and white, yes, no, but it is not. It is a scale. And that is the reality of applying such a pathotyping scheme. can be successful, but you have to keep monitoring. Okay, so I think I took slightly longer than I was allowed. I'm sorry for that. But I hope that you have now a bit of an idea of how we control nematode populations in the Netherlands, at least for potato cyst nematodes, why resistance breeding is important, and also why crop rotation is important, and also how a legal scheme, a legal reality collides with a biological reality. So if you have questions, I'm happy to answer them.
All right. Thank you very much, Mark, for the very nice presentation. Very interesting and eye-opening, I hope. So now, if anyone have questions, I would encourage students to ask first. So uh, go ahead. If you are shy, you can also chat it. Yeah. So someone is Tejo Gali. Go ahead, please.
Okay, first of all, I would like to thank Sir Iqbal for for granting us the opportunity to ask a question. I would also like to thank Mr. Mark for giving us a very wonderful and interesting lecture, if I do say so myself. So, during your lecture, I start to look at some publications. There were lots of publications regarding resistance genes. I think the major one that I found mostly is the H1 gene, but I'm actually interested in talking about the VRN gene. Now, there's this paper dated around September 2025, and the paper actually talked about the uh, the absence of cross virulence between two resistance genes, which is
Yes.
GPA3 VRN and the GPA3 SPL. The SPL
Yeah.
one is from the Solanum sparacipillum. Now,
Correct.
from a management perspective, could farmers in the Netherlands utilize a strict crop rotation alternating between potato varieties, one containing the VRN varieties and the other one containing the SPIL varieties to, I guess you could say, to reset the selection pressure because uh, I do think that at least evolution from the nematodes are possible so that they may be resistant to resistance genes. So could could Netherlands farmers, or actually just could any farmers use those two varieties in particular so that they may suppress or slow down the evolution rate to be virulence against plants with those
Yeah.
genes? Thank you very much.
Yeah. 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 Thank you for that question. Indeed, this was published by our French colleagues from the INRAE. Indeed they had two populations for Globodera pallida, which they selected on the Vernet resistance and then tested them on the Sparcipulum resistance that is very close by the Vernet one. So the agronomic reality is that the Sparcipulum resistance is not yet in a commercial crop. Actually, it was also pretty difficult to breed the Vernet resistance into a viable crop. And the reason is that potato breeders, they want to produce a good potato first and a resistant potato second. And these resistances, they come from wild potato species, so they are introduced into a Solanum tuberosum genetic background by inbreeding, and they often come with negative properties. For instance, the Vernet resistance comes with potato browning, so your potatoes turn brown very fast after you cut them, which is not something a consumer likes. So I think it is definitely a viable strategy if we have them. So your suggestion is indeed very good, and it's plausible that it would work. So academically, I fully agree. But then the agronomic practice is that we're not there yet. I also currently do not know of a potato breeder that is really far with commercializing the sparsiculum resistance so far. I hope that answers your question. Thank you.
So for now, it's I guess we could say it can't come to reality as of now.
Not yet. Not yet. No.
Okay.
That's a good idea. Good thinking.
Okay. Thank you very much for the answer, Sir Mark.
Of course. Welcome.
Thank you, Tejo, for the questions. Anyone else? Any other questions yang lain? Ada dari mahasiswa yang lain atau mungkin Bapak Ibu yang hadir juga dapat memberikan pertanyaan, dipersilakan. Boleh dalam bahasa Inggris, boleh dalam bahasa Indonesia. Kalau dalam bahasa Indonesia saya akan bantu terjemahkan. Atau juga bisa di chat, silakan. Ada dari mahasiswa yang lain? I know you have questions in your mind, just say it out loud.
Now you have me to explain.
Yeah.
If I can.
Okay, maybe while waiting for the questions from the students, so Mark, you mentioned that it is impossible practically to eradicate Globodera because we, even though we use resistance, there is always this 1% that can escape that. So then maybe some of the students also asking this question, why there is this 1%? What makes this 1% appear in the first place?
Yeah.
Do that already there from the beginning? Or they just suddenly appear when this particular potato resistance is there? So what
Yes.
happened, Mark?
For Pallida, for GPA 5 from Verney, we know it was already there. So there was just, if you had, maybe you had basics in genetics in your program at some point, like really, like with the Hardy-Weinberg, so the Punnett squares, and you have allele frequencies.
Yeah.
So we know that the GPA 5 from Verney, the virulence, it was present in a low allele frequency in the population already when it came from Peru.
Mm.
So that is definitely one of the mechanisms that, because in Peru this resistance of course also occurs in the wild, so it makes sort of sense that you'd have populations or a fraction of your population that has escaped it. The other option is that there can also be another mechanism, namely we have insane amounts of nematodes in the soil in an agricultural field. Right? I was talking to you about having like 100,000 nematodes per kilo of soil. Doesn't really matter. There are many kilos of soil in such a field. So the population goes easily into the millions. So you get random mutations, and that can, of course, also confer virulence. It's also a mechanism. Happens not to be true for Pallida in this particular case, but I can definitely imagine that it will happen at some point in nature because, it's just numbers.
Okay. Any other questions? Yes, Diva, please.
My voice.
Yes, I can hear you.
Okay. So thank you for the time. So I want to ask about besides developing the resistant plant varieties that cannot fully inhibit all the nematodes due to the complex of phenotypes that you mentioned about. Could we instead mutate the specific genes that influence or induce the nematodes to attack the plant? So is that possible? Because we know that the nematodes have a big genome. Yeah, that's my question. Thank you so much.
Yeah, thank you. Yeah, so that strategy is also a possibility. There are people trying to, for instance, express microRNAs in potato plants that then target genes in nematodes. So that is one way you can look at it. There's another thing, and namely, maybe I did not put an emphasis on this in this presentation because I think it's a very different topic, but the parasitism is very complex. So the nematode really needs to reprogram a plant cell to feed off. And it also actually needs certain plant genes to accomplish that. Basically, the nematode has sort of to convince the plant that it has to transport a lot of food towards the nematode. So another way of thinking about inhibiting can also be that if you remove certain plant gene from the plant genome, the nematode can no longer infect the plant because it misses this essential building stone. Still, it's difficult to actually achieve that. We know that there is beside our genes, there are other genes that are not so black and white that contribute, but it is genetically difficult to find these exactly. But there's definitely the whole world beyond resistance.
Yes. Is it clear, Diva? Or do you have follow-up questions?
Yeah, that's clear enough. Thank you very much.
Okay, thank you. So, Mark, we have questions in the chat.
Yes.
First question is, considering that the origin obtained comes from wild relative of potatoes, which were then crossed, what breeding strategy would you recommend for the tropical regions of Indonesia, where we use in vitro breeding and mutation for potato breeding? Okay, this first question, and then I will go to the second after the answer.
Yes. So, in vitro breeding. So let me disclaim by saying I'm not a plant breeder. I'm a nematode breeder, but I've never crossed plants. I did cross nematodes, as did Iqbal. I think it essentially works the same. So you make your mutations, and you screen for a good phenotype. And if you get a good phenotype, you can start incorporating it in your actual breeding material. I'm thinking now, do I know for successful application for this in nematode control? And then I have to say I do not know. But I have to say that also here I know that people used mutation approaches to screen for phenotypes. But in particular in nematode control, I do not know of any that was commercialized. But I think in essence, you can actually use this type of strategy also to screen for resistance.
Yeah. All right. Maybe, but if you use mutation, then it will be random. So
It will be random, and you will have less chance.
yes. So
Yeah.
it's based on your luck if you get resistance or
Yes.
no. Yeah.
And the difficulty is that nematode screenings are typically very labor intensive.
Yeah. Okay. So there is another questions in the chat. It's Indonesian, but I will translate.
Mhm.
How is the handling of pesticide in the Netherlands? Because it was said that it is banned. And in Indonesia, however, many pesticides are still being used. Also, it's not really effective against nematode. If you use crop rotation, it's a bit difficult here, especially when we have a potato industry that they need a large amount of potatoes for the industry. So but then if we use crop rotation, what would you suggest for the crop to rotate the potato? I think this is also in the context that we do not have resistant potato yet,
Mhm.
as of now. So
Yeah.
then if we rotate, then we need to use something else.
Yeah.
But then I think maybe the context of the question is that because cyst can what do you call it? can pretend in the soil for a long time, and unless there is a host, it will not hatch. What is the proper rotation plan?
That is actually difficult to answer. That is because your climate is, of course, typically warmer than ours.
Mm-hmm.
And I think, on the one hand, introducing resistance can of course help if you can if you can get it incorporated in the cultivars. The question is, what is your natural decline of the population in the soil? Because with higher temperatures, I can imagine that the decline of the population is a lot more than that we have. Because in winter, typically it's like a fridge here, so not so much going on. Whereas I think your lowest temperatures are not around 4 degrees in the soil where the nematodes are. So the natural decline might be a lot more, which is actually good news because your population will decrease faster. The situation you sketch, that there is a lot of potato has to be grown to feed the industry. It's similar to here. And that simply means that you get problems sooner. The higher your rotation frequency, like if you just keep growing potato every single year, at some point you will, if the nematodes are there, they will multiply and they will cause problems. So what actually happened in the Netherlands is that the tonnage that could be harvested per acre just was really low because you had so many nematodes. And then at some point, when the fields were cleaned up by growing resistant potato cultivars, all of a sudden farmers saw that they could get double the yield from the field compared to before. So it can have a very big impact, and farmers simply not realizing that the problem is there. Then on the metasides, they can be an effective part of control, but they also cause many side effects. One of which is that they destroy typically a lot of soil life, also diminishing the quality of the soil. So I think that is one of the reasons they're banned, because they're environmentally hazardous, and it also affects your production ultimately. But you'd have to. Iqbal has to start measuring population decline rates in soil. Then you can see what you have to do.
Yeah. First, I need to find the money. Then I'll do that.
Okay.
All right. Another question is, how can we manage so that the resistance gene won't break down under new petatypes or new virulence capabilities? And is it possible for the method to be applied in tropical countries like Indonesia? Okay. So that's
So I think in essence, resistances can be applied. There are some resistances that are temperature sensitive, so that might be something to consider. But then again, I'm not entirely sure if because you also grow potatoes in a particular condition, so I'm not entirely sure if that would be a problem. The way to manage it, that would actually be to have probably many different resistances in circulation, not all the same. So you can vary the selection pressures, as was suggested by Gali, that you have this different resistance to not have this single regime imposed. But I think it would start with monitoring. You have to know what is there, and there are typically more nematodes there than you first think. And farmers might even not be aware because they simply accepted that their fields have a low yield.
okay. Another question. Well, this is similar to the previous one. Often when we use resistant plants, it can encourage new petatypes in Globodera. Based on your experience, how can you overcome this?
Reading. Maybe an interesting twist then on the answer can be that if you have a new petotype that breaks through a former resistance, it will also allow you to find additional resistances.
Yeah.
So sort of, it gives you a new tool in the hands of a potato breeder. It's a virulent population is a new tool to screen for resistances. It's maybe nice to think about.
Yeah. Okay. Another questions. The Netherlands with subtropical region have extreme environmental condition, which is very cold and very hot temperatures in the winter and the summer. Do this condition affect the survival of Globodera? Because Globodera has the ability to forming cyst nematode. So yeah, how this environmental extreme affects survival rate of Globodera?
So what we know from it's quite well documented that in Kenya you also have Globodera infections. And what I hear from people working there is that the cysts tend to get a lot bigger than here. Still contain the same amount of eggs. Apparently, I've never seen a cyst in Kenya, but anyway, they are reported to have a similar amount of eggs, but just be much larger. And also, one of the things there is that you get multiple generations per year, because we get one generation each year. The growing season is not long enough to get multiple generations. Maybe with global warming we'll get there, but at the moment it is not. And in Kenya you have more generations per year. So that would actually mean that the problem compounds, so you get more. If you keep growing the host plant continuously, you just get more and more nematodes faster. Then again, another, as I mentioned before, I think the higher temperatures might also reduce survival rate. I know that we can best store them at low temperatures. So in that sense, our winter might enable their survival better than you would have in a tropical climate. But speculating here.
Yeah. Does the larger seas is a form of adaptation for the hot climate?
Yeah, it's a good one. We actually don't know a lot about it. We know that Pallida likes lower temperatures more. In Peru, it will it will more occur at altitudes. Rostokiensis likes warmer temperatures than Pallida. So I would expect in tropical countries Rostokiensis to be more persistently present than Pallida.
Yeah. Yeah, we do have Rostokiensis a lot in all the potato regions. Some reports say Pallida also exists, but mostly Rostokiensis.
Yeah.
But
I think
the thing is we never know what is the population density of Pallida and Rostokiensis. And yeah. Okay. Is there any more questions from the audience, from the students, or from other dermatologists? Adik-adik, bapak ibu yang lain. Oh ya, Nada.
Yeah, thank you for the opportunity. As Dr. Mark has mentioned before, that one of management strategies of PCN is the use of origins. But I don't really understand how do the researchers identify or explore the new candidate of origins against potatoes and nematodes. Do they collect a large number of cultivars globally and then screen them for resistance, or do in Netherlands they have such a long-term research, maybe for decades, on how potato co-evolute against PCNs? Thank you.
Yeah. So I think the question is, how do we get new resistances, right? We actually, in the Netherlands, as in a couple of European countries, we have a pre-Nagoya Treaty potato collection. So this is there is the potato genetic seed bank, I think that translates roughly, which is maintained actually here in Wageningen. And together with all the potato breeders, this collection is kept alive. So this center coordinates that plants are regrown every so many years, maintaining the genetic diversity. And for instance, when now we have these, because I mentioned to you, we have problems with virulence. These populations were actually also then used to screen these wild potato species for additional resistances. And that is actually then the start for getting new resistances. So what then happens is the outcomes of these experiments are communicated to breeding companies, and they can request a couple of these wild potato species for further breeding. This is a little bit different than what used to be done before. Before this pre-breeding, as we call it, basically getting to sort of a tuberosum hybrid that remotely looks like a potato you can eat. This was done non-commercially, actually, by the Wageningen research part of my university. So we are Wageningen University and Research, and the research part is an institute, and they would do this pre-breeding, and the hybrids were then made available for further breeding. But that is how it's done. It's simply screening, identifying resistance, and then that is then further inbred and characterized.
Okay.
Thank you. It was a great explanation.
All right. Thank you. Any more questions? Oh yeah. So there is one again in the chat. Until now, the way to prevent potato cyst nematode in Indonesia is by using tissue cultured seeds, and then the next generation uses rooted cutting seeds. Does this guarantee that potatoes will no longer be attacked by PCN, or we prioritize the soil health management to eradicate this nematode?
So you, instead of using seed potatoes, you use
Tissue culture.
seeds. Yeah. Well, it depends. If you have a clean field, then you probably will not take potato cyst nematodes along. But to be honest, I suspect that the main reason you get potato cyst nematodes is by farming equipment. So farming equipment from which the soil, like harvester machines, from which the soil from previous fields is not fully removed because everybody is in a hurry. And then the soil comes to a new field, and then you get an infestation. That's my guess.
Okay. Any more questions, Bapak Ibu, adik-adik, mahasiswa? Oh yeah, so there is one more questions.
Yes.
So there is NBLRR protein in the resistant genes, and I looked on the internet that it can play a role as a effector signaling secreted by the nematodes. And the question is, apakah dari? Oh, apakah dari gen yang berpotensi untuk dapat mengganggu pada penentuan? Maybe what she meant is that does the effectors or the NBRRR also involved in the, what do you call it, in the pinpointing the potential feeding site for the Globodera nematodes, like whether or not which cells they choose to feed.
The effector genes.
Yes, and also the NBRRR, is that any way interact with that? Because in my knowledge that the feeding site selection of Globodera is not yet fully understood. So you
That's
can,
great.
so we know quite a bit about how Globodera or plant parasitic nematodes modify plant cell for its feeding site, but we do not know what makes them choose particular cell. what makes them
Yes.
stop at this particular cell? So I think
Yes.
the question is, does it has something to do with effectors, or that the plant NBLRR like
Yeah.
confuse them to choose the right cell? Yeah,
Yeah.
something like that, I think.
Yeah. So first of all, it is definitely not my area of expertise. So as you hopefully notice, I know a bit about the resistance genes and virulence, but particular effectors is a whole field on itself, and it is not. Is something I rely more on my colleagues on to give a good answer. But what Iqbal says, I think, is also, to my knowledge, right. We do not know a lot about actually choosing which cell. What I do know is that the nematode, it uses, so it has this needle-like structure to probe cells, and it will look for a cell that sort of doesn't reject it. Because apparently, when it starts probing around, some cells will sort of instantly like go into hypersensitive response, like programmed cell death, and some cells won't. But like what and how, and I think people do not know. We don't even know the full effector repertoire, for instance. We have no definite answer there. So if you want to figure out, definitely do so. That will help us a lot, I think.
Yes. Okay. Next questions. Is there any transgenic plants for nematode control, like for instance BT in maize?
Not that I know of. There is not in potato cyst nematode. There is in soybean. People, I think, have engineered a soybean with multiple resistances against soybean cyst nematode. But then again, that is in the USA. Here in Europe, transgenic plants are still not allowed under law.
Mm-hmm.
We're
But
still
but is,
waiting.
but the soybean is transgenic by means that it's interested by foreign DNA, or is it breeding with
It,
HRP genes or hero
yeah, it's cisgenic. So
cisgenic?
the genes come from other soybean plants.
Okay. So it's
Yeah,
not really fully transgenic. It's a
no.
yeah.
Because it would also be very difficult, because in essence it would require you to do like a mutational screening, or at least a screening of
Mhm.
constructs for inducing nematode resistance, which nematode resistance screening is difficult, and then also having something that stops nematodes is difficult. So I think still your best guess would be to screen natural variation.
Mhm.
The way it's done, because also remember, we know for 3 genes we know the sequence, and for the rest we know roughly where they are. We don't know
Mhm.
which gene it is. If you want to do transgenesis, you need to know which gene is actually causal.
Yeah. Yeah. And while we're at it, what do you think about CRISPR or gene editing for this?
Same, right? Need to know the gene.
Yeah.
If you don't know the gene,
Say you know the gene, for instance, like
then you could, that would be great. Then you can
mhm.
have then you are get rid of this agronomical problem, where you have like a bad potato
Mhm.
that is maybe resistant, but it's not something you can sell.
Yeah.
Then you can put the resistance gene in something you can sell.
Yeah. Yeah.
That would be best. Yeah.
Mhm. So then now the bottleneck now is knowing the sequence of the genes, because so
Yes, that's the
far we only
bottleneck.
know exactly three, and the rest we just know the rough location. We don't know
Yeah.
which genes. We don't know the sequence. Okay.
Yeah.
Yeah. So
Yeah.
for my students, there's still a lot of opportunities for doing research. If you like challenge, come
Yes.
to us, Nematology.
Yeah.
Okay. So is there any more questions? No? Anyone? You can also clarify, for instance, you have a thought or an opinion that you want Mark to give a feedback on, or just clarify on something, it's also okay. Okay, I
Good.
think no more questions. So far it's clear.
Yeah, nice questions though. Thank you.
Yeah. Okay, no questions in the chat anymore, and no one raising their hand. So I think so far is clear, and yeah. So thank you so much, Mark, for giving the lecture. And before we end the lectures, usually in Indonesia we do something like to give appreciation for the speakers.
Mhm.
And at the very least, we give certificate as to appreciate your contribution for the lecture and also your time and the effort you take to enlighten us on this topic. And since we are in the very far away, I will later send you the certificate by email, but now just need to show, maybe just to show you uh, this one. So we really appreciate and
Mm.
um, and thank you for
Oh,
uh, the efforts that you make and all the
of course.
knowledge and insights you share with us as the guest speaker on this Introduction to Agricultural Nematology course with the topic of management of PCN in the Netherlands. And we hope that this is not going to be the last time you give a guest lecture for us. We hope that someday later we can also give another lectures or workshop in another opportunities. And I do hope that at some point we can meet in person in here in Indonesia. So thank you very much,
That would
Mark.
be nice.
Yeah.
Yeah.
So I will send this letter to you. Before I end the meeting, maybe something would apa namanya? Is there one or two things that want to be addressed by Bu Siwi or Bu Suryanti? Monggo Bu, kalau ada yang ingin disampaikan sebelum saya tutup, mungkin Bu Siwi atau Bu Sur.
Okay, thank you, Mr.
Ya,
Iqbal. Good night,
yes, monggo Bu.
Mas Mark Sterken. Yes, nice to meet you, even of only online meeting, and thank you for your sharing with your information for me, and I hope this information or this knowledge is can to improve the knowledge for the student. Thank you.
Yeah, you're welcome. I also hope so. That's our goal
Ya,
here to teach the
ya,
students.
ya. Thank you, Bu Suryanti. So Bu Suryanti is also the teacher in this course, Mark, along with me and Bu Siwi.
Mhm.
Uh, Bu Siwi, is there anything you would like to add, Bu? Maybe before we end the session?
Yes. First, I would like to offer apologies because I cannot open my camera. And Mark, thank you very much. Your lecture is very useful and very, for, especially for our students. Thank you very much, Mark.
Yeah, you're welcome. Glad to have done it.
Yeah.
Thanks for the
Okay.
invitation.
Oh, we love to. We're happy to. I'm happy that to have you here, Mark. So yeah, I think that's the end of the session. Again, thank you very much, Mark, for the for the opportunity to have you and for the lecture and insight that you've given us. So yeah, with this, I close the session. Thank you very much for all the students and also other audience, Bapak Ibu semua dari Nematologis Indonesia. And yeah, I hope that we will see each other again very soon in another lectures or seminar or so. All right, thank you. I close the session. Good evening, everyone. And yeah, Assalamualaikum warahmatullahi wabarakatuh. Thank you, Mark. Have a
Yeah,
nice
good evening.
afternoon and
See you
evening.
soon.
See you soon. All right. See you.
Bye.
Bye. Bye, everyone. And with this, I will close the meeting. Thank you.
Thank you for everything.
So I will end the meeting for all and yeah, have a good evening, everyone. Thank you, Mark.